my name's Ying, and this summer I worked with the Bogdanov lab on rice and tomato resistance proteins. The rice resistance protein is XO1 and tomato resistance protein is BS4. They convey resistance against Xanthomonas species through recognizing TAL effectors. However, XO1 resistance is suppressed by truncated TAL effectors known as trunk tails. What I wanted to know is how XO1 mediated resistance works, and how the differences between tails and trunk tails leads to affect XO1 mediated resistance. So what we did was that we saw that there were polymorphisms in the spine region between tails and trunk tails. So we made mutant tails and trunk tails and inoculated them into rice. What we saw is that tails with spine polymorphisms were able to escape detection by XO1, so they showed a disease phenotype as compared to resistance. And trunk tails with spine polymorphisms were failed to suppress XO1 resistance. So what this implies is that XO1 interacts with both tails and trunk tails directly in the spine region. However, when I tried a similar experiment in BS4, we found that BS4, regardless of spine mutation, is able to recognize TAL effectors and cause resistance. And it doesn't recognize trunk tails, and trunk tails do not suppress resistance. So we know that BS4 and XO1 likely have completely different mechanisms of TAL effector recognition. I would like to propose a model in which tails and trunk tails interact directly at the same region with XO1. Do you have any questions?
This is really excellent work. What was your favorite part of this?
My favorite part was probably learning all of the new molecular biology techniques that I used to make all of these bacterial constructs. I also really like seeing the results because it's very exciting seeing weeks and weeks of work pay off.
Very nice. Well, good job on all of this work. Thank you.
Thank you very much.